body has acquired immunity. It's not that that infection can never get inside your body again. Potentially it could. It's just that with these antibodies, that's going to help you defeat the infection so fast that hopefully you'll never develop any symptoms. That second round also creates more B cells, so that you're even more primed and ready to fight the infection the second time or the third time around. So first response not as drastic, not as effective, but secondary response to the same disease coming back. Hopefully you'll defeat it in record time. We saw some of the ways those antibodies can help fight infection. We saw that they stick to whatever in the body happens to have that same antigen on its surface. So they can attach to the surface of small pathogens and cover them over, stop things like viruses from getting into cells, for example. Larger antibodies, there's a kind of larger antibody that is designed to stick multiple pathogens to one another in a big clump that's easily found and destroyed by other defenders like our phagocytes. We saw that antibodies could take poisons out of body fluids, make them precipitate, so they're not dissolved and dangerous anymore. We saw that antibodies could work with complement, helping complement get a grip on a bacterium and start poking holes in it, building our membrane attack complexes. We saw phagocytes could use antibodies as handles, the antibodies opsonizing the pathogen, making it easier to grab. And at the very end, I told you that there was one kind of antibody that, instead of fighting the infection directly, would go out to mast cells and basophils, attach to them, and act as receptors against that antigen. Before, the mast cells and basophils didn't know that antigen was dangerous, didn't have the ability to detect it, so they wouldn't have triggered inflammation in response to it. But once these antibodies are installed on their surface, those histamine-releasing cells will know to trigger inflammation whenever that same disease comes back. For the most part, that's a good thing, although if the body overdoes it, that's one of the things that contributes to allergies. Occasionally, the body will launch an immune response against something that's not dangerous, just strange, like a chemical in peanuts or a chemical in seafood. And then you can wind up making antibodies against that harmless antigen, end up with an immune reaction and inflammation in response to that chemical returning to your body. Now, I'd go into a few more detail about the different types of antibodies and what they specialize in, but we aren't going to really have time. I'll just tell you that there are 5 kinds. AIGDs are the kind that are receptors on the surface of a lymphocyte. AIGGs are the ones that get sent out through the body, that look like little Ys. AIGMs are the ones with 5 Ys all attached together. The ones that attach to the mast cells and basophils are AIGEs, and the ones that get put into our surface barriers, put into our saliva, put into our tears. Yes, there's antibodies in those too. We call them AIGAs. They're like 2 Ys attached end to end. So, for instance, the antibodies that a mother gives her baby through her milk would be AIGAs. Your book shows you in a graph how much bigger the antibody response to a secondary infection is than to a first infection. So if you got, for every 10 antibodies you got in your first response to that infection, you get many more in response to the second. And I just wanted to show you this graph to clarify something. You guys saw this graph? The scale here is not linear. It's not 10, 20, 30, 40. This is 10 to the first, that's 10. This is 10 to the second, that's 100. That's 1,000. That up there, that's 10,000. So it's not 10 versus 40. It's 10 antibodies for one response. Instead, you get nearly 10,000 for the second and later responses. You can see how much bigger the higher the blue line goes. The second time around, that's 1,000 times more antibodies made, not just 3 or 4 times as many. Now, unfortunately, that's only going to work against that particular disease. If you catch another infection, like maybe a different strain of flu this season than last year, you'll have to start all over again with a primary response. same antigen on the surface as last year's did, then you will need to start all over again with a new primary lymphocyte, with a new lymphocyte that is naive in countering that disease and creating a first army to fight. Make sense? So, antibodies made against one disease aren't going to help against a different disease. You know, I wish I had time to talk about the different ways that antibodies can get into your body besides manufacturing them yourself. I'll just say that babies can get them from mother's milk and through the placenta, and in medical situations you can actually transfuse antibodies into a patient. Vaccines let you go through the primary response, the first reaction to the pathogen, without actually getting sick. They contain dead or inactive versions of the same pathogen. Like if you get a measles shot, it's got dead measles virus in it. Touching that dead measles virus antigens, that's going to teach your immune system how to fight it. It's going to breed your first army and your memory cells in response to measles without you actually having to catch them. So if you ever do get exposed to measles, your body will fight it off with a secondary response, not just a primary. Make sense? So that's the story of B cells, and that in itself is a pretty potent weapon against infection. That's not the only defense in the arsenal of the immune system, though. Besides our B lymphocytes, we also have those cells that mature in the thymus, that are created in the bone marrow, that migrate to that funny-looking gland that sits on top of your heart, like your heart is wearing a hat. That's where they get trained in how to fight infection. And they don't make antibodies. Instead, they are part of a system that sends cells, killer T cells, or cytotoxic T cells, out to search your body for infection and destroy it directly. So it's a cellular defense, much like phagocytes go out and fight the enemy directly. Now the thymus gland, we need to send these future T cells there because since they're not going to make antibodies, they don't have the ability to grow their own receptors. They can't just build antibodies and put them on their surface to detect the antigen. They need to have their receptors installed by cells of the thymus. Inside the thymus, receptors are added to each developing lymphocyte that arrives there from the bone. And those receptors, the shapes of their ends, the shapes that are going to attach to antigens and be used to identify them as such, they're generated completely randomly. All kinds of weird molecule shapes get put together there, stuck on your lymphocytes as receptors. Now that means that occasionally a receptor will be built that just doesn't work. It just won't operate. Now it's the wrong design to be a functional receptor. When you put some things together at random, sometimes you make mistakes. That's one of the reasons why each such lymphocyte, when it's had its receptor installed, needs to be tested. Let's say this lymphocyte has just had a receptor installed, only it's got a piece jutting out that prevents it from interacting with antigens. Here's an antigen. That's actually one of your own antigens, one of your self-antigens, being held up to this cell to see if its receptor is capable of checking an antigen at all. If that receptor is misshapen, if it's unable to test that antigen, it just doesn't work as a receptor, then that cell is not going to be any use if you send it out into your body fluids, or you send it out to your lymphoid tissue. It's just going to be defective. Now we saw earlier when we looked at the leukocytes that a useless white blood cell just takes up space and resources. Remember that was what was going on in leukemia. More and more useless white blood cells. That lymphocyte, once it's had a faulty receptor installed, it's not going to do any good to the body. So if it fails to recognize that that's an antigen, fails to interact with it at all, that cell's useless. And it is given an order to die. Sometimes your body finds it necessary to tell its own cells to die. We call that apoptosis. It's not always a bad thing. It's sometimes very useful in things like development. For instance, the gaps between your fingers developed by cells in between the fingers dying when you were first developing hands. In this case, apoptosis is releasing this, is removing that cell that would be no use for the body. to be allowed to travel out to the body and fight infection. We also want to make sure that the cell isn't so sensitive to self-antigens, to the antigens that are actually chemicals native to the body, that they're going to attack the wrong thing. They're going to attack our own tissues. The reason that this cell is using an antigen from our own body to test that cell is because if it turns out this receptor is too good a match for that self-antigen, then if this cell is turned loose to travel through the body, if it's allowed to leave the thymus, it's going to go find that chemical wherever that chemical is part of our body. Maybe you have type A blood, and this is a type A antigen. If this cell reacts too well to that self-antigen A, it's going to go out into your bloodstream and start attacking your erythrocytes. So an lymphocyte whose new receptor is too good a match for our own tissues, it's also told to die. Because if it's released into the body, it's going to cause what we call autoimmunity, self-immunity, the immune system attacking itself, attacking our own body. There's a lot of autoimmune diseases you may have heard of, like MS, type 2 diabetes, lupus, cases where the immune system mistakes our own tissues for a threat. To try to prevent that from happening, we're going to check that that cell is not too good a match for our own antigens. That's not going to go out there and try to attack our tissues in the belief they're germs. What we want is for the receptor to work, but not to match any of these self-antigens that are used to test it. They're inside the thymus gland. Only after the thymus cells have checked that receptor thoroughly and found that it is functional, but that it is not sensitive to our own body chemicals, will it be turned loose from the thymus gland to travel out through the bloodstream and eventually to our lymphoid tissue. At that point, it's become a mature, immunocompetent, although still naive, T cell. Make sense? Okay, questions so far? All right, so this T cell has met with the thymus gland's approval. It has working receptors, and it's not going to be a threat to our tissues by attacking our own body chemicals. What exactly it's going to do next depends on which of two families of T cells it belongs to. There's two versions of the T cell of interest to us. There's what we call a CD8 cell. That's a cell that is going to be the parent of fighters, of cells that will actually go out and physically attack intruders. These are likely the lymphocytes you saw under the microscope when you looked at blood in the lab, because they spread all over the place, searching for virus-infected cells to attack, searching for cancer cells to attack, searching for anything that has an antigen they recognize with their newly installed receptor. The other category of lymphocyte that leaves the thymus is the CD4 cell. This is going to be the parent of cells whose job is not to fight, but to assist other defense cells, what we call our helper T cells. Helper T cells, their job is to regulate the action of the rest of the immune system, to help tell B cells that it's time to recognize antigens and make antibodies, to help CD8 cells recognize